Hello, my name is Bas Thiel, and today we're going to talk about assembly strategies for genomics and metagenomics. We use sequence assembly to combine DNA sequences that are derived from a DNA sequencing platform into longer sequences that may get closer to representing the genome of organisms that we are interested in. So DNA sequencing I discussed in another lecture, this one over here. You can find it on YouTube. And so most DNA sequencing platforms actually yield reads that are shorter than 1,000 nucleotides. So both first generation sequencing, which is Sanger sequencing, that has existed since the 1980s, as well as second generation sequencing, which is the massively parallel sequencing, both read relatively short read lengths. Longer read sequencing platforms are emerging. PacBio and Oxford Nanopore are currently the ones in the market. Although they identify longer sequences than the first and second generation sequencing platforms, still these sequences are, or these reads are not enough to cover the full genome. So we will need to use sequence assembly to identify, to combine these sequencing reads into longer contigs, longer sequences. I'll tell you about contigs in just a second. So people have been using computers to combine sequences into longer sequences since the 1970s, and there are two basic strategies. The first is to use a reference genome. This is called reference-guided assembly. And the second is to use is a data-first approach, which is called de novo assembly. So if we have a reference genome of a very similar organism, so something like the same species available, then we can take that reference genome and map the sequences that were, or the reads that were sequenced by the DNA sequencer to this reference genome and identify the consensus nucleotide at each of these positions and infer a consensus sequence that represents the information in the sequencing reads rather than in the reference genome. Of course, the reference genome may have regions that are not present in the sequenced organism, and so those parts will be missing and might lead to broken sequence of the genome of the organism we're actually interested in. Of course, the closer the reference genome is related to the sequenced organism itself, the better this reference-guided assembly will be in the end. Okay, the second approach is de novo assembly, and in de novo assembly we don't use a reference genome, but we use only the data itself and look for overlaps between the DNA sequencing reads, which after identifying are then combined into longer sequences like this. For reference-guided assembly, we need a closely related reference genome. This is necessary so that we can align the sequencing reads from the organism that we're trying to assemble to this reference genome and reconstruct a consensus sequence. Preferably, the reference genome is from an organism of the same species, but if it's more distantly related, the probability will increase that we will have structural mutations in the genome, for example, insertions or deletions or rearrangements. And because the sequencing reads are generally short, especially, of course, for second generation DNA sequencing, the short sequencing reads might be mapped to positions in the reference genome that they do not actually belong in the organism that we're trying to assemble. So if we have a more distantly related reference genome available, then we would also need to be more permissive when aligning the sequencing reads to the reference, because of course also point mutations will have increased at a larger phylogenetic distance, and this could lead to ambiguities in where the sequencing reads are mapped and potentially to errors in the reconstructed genome sequence. So reference-guided assembly generally is not considered to be appropriate for identifying or for assembling actually sequences of completely new species. And as we know from metagenomic studies, there are many species, bacterial species, that we have not yet seen before. So this is a view of the tree of life generated in 2016 from complete genome sequences. as well as genome sequences that were assembled from metagenomes. And as you might or might not be able to see, there's very many of these clades that are not very well characterized. So the italic printed clades are the ones that are well characterized, and the rest are have not yet been seen before, and so these represent clades where we definitely do not have suitable reference genomes available for reference-guided assembly. I will get into how we deal with that later on in this presentation. So first I want to give you some overview of the terminology. So there's the terminology for a DNA sequence assembly. So of course there's DNA. DNA is the double-stranded helix that we see over here. And after fragmenting in the lab, these DNA sequencing, the DNA sequence is fragmented into DNA fragments, and each of these fragments is then loaded onto the sequencing platform, and I'm talking now about second-generation sequencing, and then forward and reverse reads of this DNA fragment might be identified. Depending on the length of the fragment, and this is something that you can tune in your experiment, the forward and reverse reads might overlap in the middle or not. So this is, this depends a little bit on how the experiment is set up. Generally, these fragments might be a few thousand nucleotides long, and the DNA sequencing reads might be a few hundred nucleotides long, so then they do not overlap here in the middle. Of course, we are going to sequence several millions of these DNA fragments from the forward and the reverse end, and so many reads will be identified. And when we look for the overlaps between these reads using de novo assembly, which I'll discuss in just a few minutes, we will be able to identify contigs. And contigs represent sequences that are contiguously represented by sequences in the DNA sequencing reads. So here the DNA fragments and reads are sorted by their position on the genome that we're trying to sequence. And as you can see, at every position in this genome that's going from left to right, we have information about the nucleotides present in the sequence data. So the sequencing reads cover all of this region over here. Here there's no information for a little bit, and then again there's contiguous information in the sequencing data in the reads. Here there's another gap of the genome, and then there's another couple of reads that are covered. And if we assemble all of these DNA sequencing reads together, we get a contiguous sequence in this stretch, and in this stretch of the genome. There's two little gaps that are not present in the data. And these gaps are a little bit different because for this gap we actually know that there is a link because the forward and reverse read sequence from the same DNA fragment are present on different contigs. Here we do not have that information, so it's not clear whether this contig, this piece of sequence over here, should be left or right of the other contigs in this genome sequence. So this information, the fact that these two contigs are side by side basically in the genome, that can be represented in another level of assembly, namely the scaffolds. So in scaffolds, contigs are scaffolded together, and regions that we know are there but we don't know the sequence of are represented by ends. Ends are basically characters that represent any nucleotide, A, C, G, or T. And this information might be, the information that these two contigs are side by side on the genome, might be represented either in the information from the forward and reverse DNA sequencing reads, but it might also be information that we take from a reference genome again. So if on a reference genome these two contigs map side by side, then we can assume that in the genome that we're currently trying to sequence, if the reference genome is close enough, of course, these two contigs are also present side by side. And we can represent that by including these two contigs into a single scaffold. So multiple scaffolds then, this is the last step of assembly, of genome assembly, and this is specifically important for metagenomics. Multiple scaffolds might be combined together into bins. And so bins represent scaffolds or possibly contigs as well that are derived from the same genome. And in a metagenome, of course, all of these might be mixed up because there's multiple species that are sequenced at once. And so we want to combine these sequences from multiple organisms into different bins representing the same genome. So this terminology is important, and I will use these terms throughout the presentation. Another term that I want to quickly introduce is coverage. So coverage is an ambiguous term, so watch out when people use the word coverage and think about what they're actually talking about. So coverage could mean two things. Either it's the number of reads that cover a nucleotide in the assembly. So if we look here at the genome that we're trying to assemble, and these black lines are the sequencing reads that we have assembled at these positions, then the coverage would be the number of reads that cover a single nucleotide at a given position. And another term that coverage that's also used, where coverage has also been used, is the percentage of the target genome that has actually been recovered after sequence assembly. And so this is basically in the horizontal direction, how much of this reference genome has been assembled. And so, for example, here there's a piece that's not been assembled. So for this target genome, I guess about 90% of it or something would be recovered. So this would be a horizontal coverage of 90%. The vertical coverage in this region would be lower than in this region. Okay, so this is an ambiguous term, so of course we try to resolve that by using different words. And the vertical coverage is also called depth. So depth is the number of times that a given nucleotide in a target genome has been sequenced or has been recovered. The horizontal coverage is the term that we use for the horizontal coverage, and this is the percentage of the target genome that has been recovered in the assembled contigs or scaffolds. What's interesting or important to remember is that the depth or vertical coverage is actually also representative of the abundance of the DNA sequence in the original sample. Because the sequencing reads are derived from DNA fragments, and these are randomly sampled from the original DNA fragments, the abundance of the DNA actually is represented in the depth of an assembled contig. So this contig over here is actually present more abundantly or more frequently in the original data than this contig over here. So this might, for example, be representing a repeated region in this genome that has been assembled into a single contig. So we'll get to that in just a second. Okay, so now that we know of all of the terminology, let's continue to the assembly algorithms. So the first, there's one thing to mention, which is that for de novo sequence assembly, we need enough data. Of course, we need sufficient horizontal coverage of the target genome. All of the nucleotides should actually be present in the sequencing reads, otherwise we cannot assemble it. But we also need enough depth. Because if there's not enough depth, then we might have an assembly program might not be able to identify these little overlaps between two sequencing reads. So we need enough data to be able to assemble a genome. It's also important to realize that de novo assembly breaks on repeated regions and on low coverage regions for the reason just mentioned, but also on repeated regions, and this will be explained in a little bit later. And then there are three different de novo assembly algorithms that I want to discuss. The first is the greedy assembly algorithm, which I will only use to illustrate certain concepts and ideas. The second is the overlap layout consensus algorithm, and the third is the De Bruijn graph. So the greedy assembly algorithm is very simple. It has 4 steps. In the first and second step, you take all of the sequences in your data set, which initially are the reads derived from the sequencing machine. You identify similarities between every pair of sequences in the data set, and then you find the best matching pair to assemble. So these two sequences are combined and assembled into a single contig. So this is your first contig. The two sequencing reads that you assembled can then be removed from the original data set, and the contigs can be added to this data set And by iterating these 4 steps many times, you should, in an ideal case, of course, end up with a single contig representing the genome that you sequenced and that the data is derived from. You can imagine that this could work well if you have not too many sequences in your data set, and if the sequences that you are comparing are quite long. So if the longer sequences would give a clear best match in the third step of the greedy assembly algorithm. And this best match would allow those sequences to be easily assembled. But if you don't have very long sequences, you might get multiple best matches. So this is going to be a problem. And also, if you have many sequences, such as in a second generation sequencing data set, where you have these millions or sometimes even billions of sequencing reads from a single sequencing run, the all versus all comparisons that you want to do in step 2 of the greedy assembly algorithm might become very computationally expensive. And so this is actually not a very good approach for de novo assembly of next generation and second generation sequencing data. Then there's another problem, which is also important, given that much of the sequences that we're trying to assemble are actually relatively short, which is a problem of repetitive sequences. So if in our genome sequence, which we've indicated here once again, there's a repeated region, so this could be a gene that has been duplicated in the genome, it could be a transposon or something like that. And this repeated region has been sequenced by across with 4 sequencing reads. So there's these reads A, B, C, and D. Then if we would apply the greedy assembly algorithm, then the first 2 sequences that would actually be assembled would be A and D, because they share this large region that's present in the repeat. So this repeated region would be identified by the greedy assembly algorithm first. A and D would be merged into a single contig, and then after that, the only overlapping part that could still be identified and assembled is the tails of B and C, which would then be assembled into another contig, and we would end up with having 2 contigs rather than a single contig representing the entire region across. So this is of course wrong, and to avoid this, we would actually need to look at all of the possible ways that the reads A, B, C, and D could be assembled into contigs first, before actually joining any 2 reads into a contig. So to do this requires actually a global view of all of the possibilities, and this could be achieved by using a graph-based approach. So a graph is actually, so for example here we have a graph where we have all of the overlaps indicated by dotted lines. And so A overlaps with D indeed, which is the one that is assembled together over here. But there's also an overlap between A and B, between B and C, and between C and D. And we would be able to, by taking this graph approach, we would be able to identify a path through this graph that represents all of the data, and then we could, if we took this graph approach, assemble everything into a single sequence, so a single target contig. So assembly algorithms that use this graph approach, not specifically this graph approach only, but the graph approach in general, are more successful at dealing with real biological data. And a graph, just to recap, a graph is actually a structure, a mathematical structure that contains nodes and edges. Nodes are, could contain something, and edges contain connections between the nodes. And there's 2 types of graphs that are often used in sequence assembly. One is the overlap layer consensus graph, and the other is the De Bruijn graph, which is named after the Dutch mathematician De Bruijn, Dick De Bruijn. I will discuss these both. So first is the overlap layer consensus graph. This is the, I guess conceptually the simplest graph-based assembly approach. So what this does, if we have the genome sequence here at the top, then we have 5 sequencing reads, J, K, L, M, and N over here. Then what is done in the overlap delta consensus graph is to identify the overlaps between each of these different sequencing reads. So the nodes in the overlap delta consensus graph represent reads, and the edges represent overlaps between the reads. So we can see that J overlaps with K, J also overlaps with L over here, J overlaps with M, J does not overlap with N, so there's no edge from J to N, and then K overlaps with L, K overlaps with M, and K overlaps with N, so there's all of these edges that are drawn in this graph. So what we're looking for is this path that passes every node once, and this is called a Hamiltonian path. This is a concept that has already been known from mathematics for a long time, or from graph theory. And it's known that finding a Hamiltonian path is actually a difficult problem for computers. It's so-called NP-complete. And this means that for very large graphs with many nodes, such as potentially derived from high throughput next generation sequencing data sets, are actually extremely computationally intensive to resolve. So there's no efficient algorithm available, and this is what makes overlap delta consensus less suitable for data sets with very large numbers of sequencing reads. So once the Hamiltonian path has been found, the consensus sequence at every position in the genome sequence can be determined by looking at the nucleotides represented in the reads. And together, this completes the overlap, layout, and consensus algorithm for de novo sequence assembly. So the De Bruijn graph is an approach that is suitable for large-scale sequencing data, such as derived from second-generation sequencing, where we have millions or sometimes even billions of sequencing reads. And the idea of the De Bruijn graph is that we create a graph not of reads, where the data reads is represented in the nodes, but we create a graph where the reads are actually represented in the edges of the graph. And we do this as follows. So every read is split up into shorter words called kamers. For example, if we look at read M over here, we can split it up into kamers of length 29 nucleotides as follows. So there's, well, let's say about 15 or something kamers of length 29 in this read M. And you can imagine that if we're dealing with a genome sequence, so if all of the reads are derived from the same genome, then the number of kamers in the graph will never be larger than the number of nucleotides in the genome, because at every position in the genome, a new kamer will start. And so, for example, for a bacterial genome, we will expect at most a couple of million kamers that start at every position. So like I said, the nodes in the De Bruijn graph are these kamers, and then the edges of the graph are actually the data itself. So the reads are represented in the edges of the graph. So if you look at the graph like this, these are all of the nodes, and then the edges in between are actually the information in the read. So if two kamers are adjacent to each other in the data, so in the actual read data, then these kamers are linked by an edge. And so the edges can be weighted by the number of reads where these kamers are found to be adjacent. So if we can imagine this representing a single genome assembly, then we can see that a very simple graph will actually arise where all of the kamers are nodes, and if there's a simple genome where there's no repeats, for example, in the genome, then there would be a single path that connects all of the nodes, and all that we need to do is find a path that connects all of the data, in this case all of the edges, and this is called an Eulerian path. So the Eulerian path is actually very easy to calculate for a computer. But of course in practice, and especially in metagenomic data sets, it's not so simple, because there are many parts of the data that are present multiple times. So there are many repeated regions in the genome sequence, or there might be shared regions between different organisms in the metagenome. And this will lead to certain k-mers being present multiple times in the target sequence. So for example, here we see a region in the graph where certain k-mers have to be visited twice to be able to represent all the data in a single graph. So this means that there is a region over here in the genome that is repeated, and these k-mers are present multiple times. This can be partially resolved, and different assembly algorithms actually use different methods to do this. Can be partly resolved by using the width of the edges, which, as you remember from the previous slide, actually represents the number of reads that are linking these two k-mers in the data. And the number of reads is directly proportional to the frequency of this piece of DNA in the original data set. So if a given piece of DNA is more frequent, that means that it's probably present repeatedly in the target genome sequence. So like I mentioned, there are several different algorithms available. The first popular algorithm was already from 2001 to do this, the Brown graph assembly. But since there's many algorithms available, at some point we need to try to assess what, which ones are the best ones. So if we do a de novo assembly with different programs, how do we assess the quality? So there are several approaches to do this, and so for example you can look at a very simple measure, which is the length of the longest contig. So if you're assembling a single genome, you would expect that ideally all of the sequencing reads would be assembled in a single contig. There's also measures like N50 and N80, I'll explain those on the next slide, and they're also derived from the distribution of sequence lengths of the contigs. But none of these are very biological measures. So if you would stick together all of the reads in a sequencing data set end to end, you would get a really, really long contig, but it wouldn't make any biological sense. So then there's also some other measures, for example, the percentage of paired end reads that are matched when you map them back to the assembly. So as you remember, paired end reads are derived from the same DNA fragment, so the left and right side of a certain DNA fragment are sequenced together, and if you align these sequencing reads back to the assembled contig, you should expect that the read pairs, the forward and the reverse read from a given pair, should be aligned to the same contig and in the right orientation. So the percentage of paired end reads that are properly matched is a measure of assembly quality as well. An even more biologically meaningful measure is the length distribution of open reading frames. The open reading frames in a random sequence are statistically very unlikely to occur because 3 in every 64 codons should be a stop codon. So there's, if you find open reading frames of the length of a real protein sequence, this is an indication that the sequence represents a real biological sequence, while a random sequence would have much shorter open reading frames on it. Okay, so then there's N50 and N80. These are very often used assembly length statistics, but don't just look at the length of the longest contig, but take the distribution of contig lengths into account. And they work as follows. So if you take all of the contigs and place them end to end, starting with the longest contig over here and then the shortest contig at the very end, then you get a long sequence of nucleotides, and the N50 length is the length of the contig which is present at the 50% mark. So basically the contig in the middle if you place them end to end. It's not the same as the length of the middle contig, because the length of the middle contig could be much shorter than the N50 length. Similarly, the N80 length would be the length of the contig at 80% of the cumulative length, and you can come up with any number of, or any n number, and this, of course, as the n number increases, the length of the contig at that position further decreases, as you can see here, because they're sorted from long to short. So these measures are used, and they're of course very good to assess the quality of a single genome assembly, where you expect a single long contig to be reconstructed by the assembly program. But for metagenomics, it's maybe a little bit less meaningful because metagenomes might be derived from highly diverse communities, and some of the genomes in the metagenome might not be fully sequenced, so you cannot expect to reconstruct all of those in a single context. So the length statistics might be a little bit yet less useful for metagenomic assembly. Okay, so here's an overview of some of the structures that are present in the Bruin graphs, or that might be present in the Bruin graphs. We've already talked about the cycles, and this is a region in the genome that represents a region in the genome that's present twice, a repeated region, on the same context. So this path goes through this region twice. Then there's structures such as frayed ropes, bubbles, and spurs, and they all represent different types of problems or things that need to be resolved by the assembly programs. But I want to go a bit deeper into the repeats specifically. So imagine we have two sequences in our DNA that are repeated. For example, a gene that is shared between two different genomes. Then the difference between the overlap layout consensus and the Bruin graph could look like this. So in the one, the nodes in the graph are the reads, and the overlap are the edges. Sorry, are the overlaps between the reads. And the repeated region could look something like this. So there's a structure over here where we cannot resolve whether the green graph part of the graph would continue into the blue part, or whether the green part would continue into the orange part of the graph. Similarly, in the Bruin graph, the graph would look like this. The nodes are the k-mers, and the reads are the edges between the k-mers. But here as well, we have a certain part in the graph where green and yellow merge, and then it's unclear whether green continues into blue or whether green continues into orange. So most assembly programs, most de novo assembly programs, would actually, in such a situation where they don't know what to do, cut the graph into parts and say that there's 5 different contexts that are being assembled: a green context, red, blue, yellow, and orange context that are being assembled in all of these cases. And so it is unlikely that in this situation the assembly program would actually lead to a chimeric sequence where the genomes are wrongly reconstructed. So green would continue into orange, for example. This could only be the case if the genomes are not very deeply sequenced. And for example, the blue part of the genome has not been sequenced by reads, and there's only this green part over here and the orange part over here present in the data, for example. Then you could get a chimeric sequence arising that combines different sequences from different genomes into a single context. This is of course more likely in metagenomic data sets. But it's not just in metagenomic data sets. So this problem is also present in single genomes. So for example, the 16S operon, or the ribosomal RNA operon, including the 16S gene, is actually present multiple times in many bacterial genomes. For example, Salmonella has, I think, 7 of them. Interestingly, it's not just in bioinformatics that we would not be able to resolve this path through this graph over here, but it's been shown already in 2003 that Salmonella actually recombines its genome at these different RRN operons. So there's different strains of Salmonella that have different paths through this sort of genomic graph that they've created by recombination at the RRN operon. Of course, this problem becomes much more pronounced in metagenomic data sets where there's many different strains of the same species, as well as different species from the same genus, etc., present, all mixed up together. And this is what makes metagenome sequencing or metagenome assembly much more complex than genome assembly. Okay, so let's continue about metagenomics right now. So this is an example of a picture of how important the human microbiome is. But there's, of course, microbiomes everywhere. There's the ocean microbiome, and in many different data sets, and many different biomes. are available from environments around the world, including soil, fresh water, wastewater, etc. And all of these data are available in public resources such as Magnify or the NGVAS database. And we still want to assemble longer contiguous sequences of the microbes present in all of these environments. So if we compare genome versus metagenome assembly, then we've already discussed a little bit how we expect a single sequence to emerge from a genome assembly, depending of course on the coverage of the genome. Do we have enough data to represent the full genome sequence and assemble it? This sequence should be contiguous, especially if we're dealing with a bacterial sequence that contains only a single chromosome. Then we expect one sequence to arise from the assembly, and it should be reasonably evenly covered. So the length of the genome should be covered by the same number, more or less the same number of reads, leading to a stable depth profile along the length of the contig. In contrast, in metagenome assembly, depending on the diversity, we will expect many different sequences to be assembled because there's many different organisms present, so there's many different genomes that the data are derived from. And the sequences are thus also fragmented. So some of the organisms might be very abundant, leading to a very high read depth, and some might be quite rare, leading to a much lower read depth in the data. In genome sequencing, we are mostly dealing with clonal sequence. So all of the reads should represent the exact same genome sequence and should be able to be assembled in one sequence. Whereas in a metagenome, there's a lot of natural microdiversity. So there's a lot of mutations in the genomes of closely related strains. And so in metagenomes, we don't know if a mutation observed in the reads is actually a sequencing error or naturally occurring diversity. Whereas for genome sequencing, we will probably we can be quite sure that low abundant mutations in the reads data are actually sequencing errors, and we can ignore them. Then the final important difference between genome and metagenome sequencing is the presence of repeats. So I've already illustrated repeats previously. In genomics, repeated regions consist of duplicated genes or conserved domains within the same genome. But in metagenomics, repeats could also include, like I mentioned before, closely related strains or horizontally transferred genes between different organisms. And this leads to a much higher complexity in the assembly graph, and they are much more difficult to resolve. And in the worst case, for example, when two genomes that share a certain gene region are both very lowly abundant, it is possible that you miss parts of both genomes and that you could create chimeric sequences. So this is the big fear of all of the people working on the metagenome assembly, is don't we get chimeric sequences in our data. The chimeras are, of course, organisms that are composed of parts of different organisms, and these are, if you want to make a good biological interpretation of the sequences that you've assembled, they are very bad. You don't want them in your data set. So how big is this problem of chimerization really? Well, I've already mentioned that good assembly algorithms should actually break contigs at ambiguous sections, like here. So we have a blue genome and a red genome with a shared gene here in the middle. If the contig graph, either overlap layout consensus or the Bruin graph, encounters such a shared region, then generally what it will do is it will create 5 different contigs and say, I'm not sure how to actually resolve this part of the graph. If you're still afraid of chimeras, maybe the following thoughts are helpful as well. So chimerization is probably going to be more frequent between closely related strains because closely related strains will share more of their sequence, and they're very similar sequences. And so if you have very closely related strains that do end up in a chimeric contig, then perhaps the biological impact or the impact on the biological question is not as big as if those chimeric sequences were derived from very different organisms. So for example, here we see a chimeric organism. This is a real one. This is Venus, the amazing chimera cat. She has a Facebook page and everything. So she is a real chimera, but these are very closely related strains that chimerized, and so we know what this organism will do in the environment. So it's very easy to give a good biological interpretation of the role of this organism. So perhaps with closely related organisms, chimeras are not such a big problem. You can also estimate the effect of chimerization on your biological result by using a bioinformatic approach where you use different stringencies of assembly. So you could use a high stringency of assembly where you expect very few chimeras to be created in the assembly. And you can use a lower stringency where you assemble more divergent sequences into a contig, and you will expect more chimeras to arise in that situation. So if you compare the biological result by using these two different parameter settings, then you will be able to assess the effect of your chimeric sequences on the biological interpretation, and basically place these chimeras in sort of a little cage and assess how big their effect is on the final result that you're trying to describe. Then finally, there's another approach which you can do to look at chimeras, and this is by assessing actually the depth profile along the length of the contig. So here we see a contig at the bottom, going from left to right, and the number of reads that are mapped or aligned to each of the positions on the reference genome, or on the, sorry, not on the reference genome, on the assembled contig are indicated with these gray areas. And you can see that there's quite a sharp drop in the depth along the contig length, indicating that this might be a chimeric contig that was assembled, where this part of this organism actually is much more abundant than this organism over here. And this contig, we should probably break this contig somewhere over here. This image is actually derived from the website of the Marin Lab, which has a very nice description of coverage, which I recommend to read. Okay, so if we continue wondering how to assess assembly quality for metagenomic sequences, metagenome assembly specifically, there are several additional metrics that we can use. So we've already discussed the length of the contig and the N50 and the N80 values, the percentage of matched paired end reads that are aligned to the that can be aligned to the contig. We've discussed the length distribution of open reading frames. But specifically for metagenomics, there are some other metrics that we can also use. And this is the percentage of the data that is included in the assembly. This is an important measure because for a metagenome, we want the assembled contigs to well represent all of the data that is present in the original metagenomic sample. So we want to represent the community, the microbial community well. We can assess this by aligning all of the sequencing reads back to the assembled contigs and just measuring how many of these reads can be aligned back to the assembled contigs, indicating what percentage of the original data is actually explained by the assembled contigs. So do the contigs actually explain the original data well? And finally, the evenness in depth along the contig length is also something that is supposed to be quite even. So this is for metagenomics also a good way of assessing whether proper contigs were assembled. So now we've assembled some contigs. We've assessed which algorithm is the best to assemble these contigs with. We've calculated some assembly statistics. We know that this is the best contig assembly that we can get. What is the next step to take? Well, in metagenomics, it's possible that each of these contigs is derived from a different bacterium, but it's actually more likely that these contigs represent fragments of the genomes of different bacteria, and that some of these contigs actually belong together in the same bacterial genome. So in the next step for metagenome assembly, we do a step that's called binning. And in binning, I try to identify the contigs that are derived from the same bacterial genome and place them together in a bin, which represents at least a better representation of the complete bacterial genome sequence than the separate contigs. So to do this, one of the signals that we could use is the forward and reverse read that are the forward and reverse read pairs that were sequenced. And this is a process that is known as scaffolding, which actually we've discussed all the way in the beginning of this lecture. But often the read pair information is already used when creating these scaffolds. So instead of contigs, we could also try to bin scaffolds. So we're really looking for properties of the bacterial genomes, so properties of the sequences that are stable along the length of the genome, so that we can identify those properties in the assembled contigs and try to use those to link the contigs together that belong together in a single bacterial genome. So one of the properties that is often used for this is the GC content. So GC content is quite stable along the length of the bacterial genome, and it's also quite different between different bacteria. So here we see a graph that has lots of little colored dots. Each of the little colored dots is a contig, and they are linked together in this graph by matching forward and reverse read pairs. So in this case, apparently those read pairs were not used for scaffolding, but for linking the contigs in this graph. And what you can see is that all of the contigs that have been linked in this graph tend to have the same color, which means that they have the same GC content. And different edges in this graph, or different stretches, pieces of chromosome, have different colors, meaning that we can also distinguish different bacteria from each other that have a different GC content. So this is one of the properties that we could use to identify contigs that belong together in the same bacterial genome. This has actually been taken to a higher level by not looking at the GC content, but looking at a Kamer profile. And Kamer profile is basically a sort of a higher dimension version of the GC content, because instead of just looking at the G and C content of the sequence that we're looking at Kamers, and then the Kamer percentage of a sequence. So for a given sequence, we can divide it into Kamers. We've already seen this with the approach to construct the Bruin graphs. But we can also count the percentage of each of these Kamers present in the graph, or in the contig. So this is a characteristic that is quite typical of a given bacterial sequence, because bacteria tend to use a certain type of nucleotide usage along the length of their genome. For example, in this sequence here, there's a lot of ATT and a lot of GAT, but less TGA, TTC, and TTG that have been used in the sequence. And if we make such profiles of Kamer abundance for each of the different contigs, then we see that those are actually quite similar between different contigs from the same bacterial genome. So as you can see, this is sort of very this is the same actually as the GC content, because in the GC content we just count the percentage of G's and C's versus the percentage of A's and T's, which is a very low-dimensional, one-dimensional version of the Kamer profile. So the Kamer profile actually contains more information and allows you to distinguish more different types of bacteria like that. The length of K that is often used in metagenomic binning is 4. So these are called tetramers or tetranucleotides, and such a profile is then also called sometimes a tetranucleotide frequency profile, or TNF. If you plot such a tetranucleotide frequency profile in a PCA plot, principal component plot, you see that contigs, in this case each of the little dots is a contig, from the same bacterial or archaeal genome actually cluster together in this multidimensional space. And this is, and you can identify these clusters with a similar tetranucleotide frequency profile as being derived from the same genome. And you can just use this to cluster together the contigs that are derived from the same genome and bin them. This is an approach that was already introduced in 2007. Another signal that is consistent along the length of the bacterial genome is the abundance, which can be represented as the vertical coverage or the depth of the assembled contig. So if we assume a bacterial community over here with different bacteria in it with different abundances, then if we sequence this randomly, then there will be different DNA sequencing reads sequenced from this community, and the red bacteria will be much more abundant than, for example, the purple and the yellow bacteria over here. And after assembly and mapping the reads back, we can identify this depth for each of the different contigs. And so by looking for contigs that have a similar depth, we can actually cluster contigs that are derived from the same bacterial genome based on the original abundance in the sample. Of course, there's some noise in this due to the sample prep and due to the sequencing chemistry. Some parts of the genome might be more frequent in the final data set. So let's look at what this really looks like in a real example. So here there's 19 contigs that have been binned based on their depth in 3 different samples. And you can see that there's quite some variability, but on average, each of the contigs has a similar depth in samples 1, 2, and 3, meaning that in sample 1, this organism is more abundant than in sample 2, and in sample 3, it's slightly less abundant than sample 2 over here. So you should consider that this is it's quite stable along the length of the genome, and this can be used to bind together these contigs from the same organism. Here we've actually looked at the depth in 3 different samples. So this is once again a higher dimensional version of the abundance signal. And yeah, if you have metagenomic sequence data available from multiple samples that have the same organisms in them, then you can use a depth profile across multiple samples, in this case 3 samples, to get a higher resolution image and better separate out different bacteria that might have a similar depth in just 1 or 2 of the samples. So the more samples you have, the more variability there is in the abundance of the different bacteria, and the better you will be able to separate out the contigs from different bacteria into separate bins. So then the question is, how do you get different samples, different metagenome samples or data sets, with different bacteria in, or with the same bacteria in them, but at different abundances? Of course, you could sample the same microbial community in different locations, but you can also use a different approach, which is to sample the same environment, and in this case, for example, a bioreactor. You could sample it at multiple locations within this bioreactor. You expect the same sort of microbial community to be there, but maybe higher up, which is further away from the oxygen that's being bubbled in this bioreactor, there might be slightly different abundances of the different bacteria in there than at a lower point. So then you can sample these 2 locations, but you can also treat the samples in a different way. You can, for example, in this case, there's floccular biomass and there's granular biomass that are present in the bioreactor. And you can separate these out. So in both of these types of biomass, similar microbes will be present, but at different abundances. And then you can also influence the amount of DNA isolated from different bacteria by using different types of DNA extractions. So here we used organic extraction and the power soil kit, which is another approach for isolating DNA from metagenomic samples. And different bacteria have a different sensitivity to these different types of extractions. So all in all, we have 2 different sample points, 2 different sample treatments, and 2 different types of DNA isolation, leading to 8 different metagenomic data sets, in each of which the bacteria which are present in the original sample are expected to be present at different relative abundances. So some might be more present in this one, and some might be more present in this one. And this allows us to get a depth profile of every contig across 8 different samples, and this depth profile will allow us to very sensitively bind together the contigs that belong in the same bacterial genome. This is basically the story of binning. So there are several tools also available to do this. And once again, if we have multiple tools or multiple parameter settings, we will want to know which is the best one. So how do we assess which binning approach yielded the best or most complete bacterial genomes from a metagenomic assembly? And for this we use single copy marker genes. Single copy marker genes are genes that are present only once in all known bacterial genomes. So this is based on the genome sequences of bacteria that we have available in the database to date. And there's about 100 genes, most of them are ribosomal proteins or encode ribosomal proteins, that are present in all of these different bacteria. And what you can do is when you create metagenomic bins, which are supposed to represent complete bacterial genomes, you can count how many of these single copy marker genes are present in the bin, and by doing so you can estimate a percentage of completeness of this bacterial genome. If certain single copy marker genes are present more than once, that is unexpected, so you've probably added too many contigs to your genome bin. And for example, these contigs could represent sequences from a related strain that have similar binning signals, so similar gamer profile and similar abundance profiles along samples. But you've you've added redundancy to the genome bin, and thus you can use single copy marker genes to estimate both the completeness but also the redundancy of metagenome assembled bins. Okay, so this completes the lecture about assembly strategies for genomics and metagenomics. Yeah, that was it. Thank you very much.